Welcome back to a new video session from Biology Boost Up, and in this video session we're going to study about the heartbeat. Okay, so now let's start. Now at first, what is heartbeat? To pump the blood into blood vessels, the heart regularly contracts and relaxes in a rhythmic manner. It is known as the heartbeat. The heartbeat includes one systole or contraction phase and one diastole or relaxation phase of the heart to distribute and receive blood to and from body. Now let's talk about the types of heartbeat. The heartbeat is two types. First, the neurogenic, and second one is myogenic heartbeat. In case of neurogenic heartbeat, it is initiated by a nerve impulse coming from a nerve ganglion situated near the heart. It is present in some annelids and arthropods. On the other hand, in case of myogenic heartbeat, it is initiated by a patch of modified heart muscles itself. It is found in hearts of molluscs and vertebrates like humans. Now look at the origin of heartbeat. First, as the mammalian heart is myogenic, the heartbeat originates from a muscles. However, it is regulated by a nerves. The heartbeat originated from the sinoatrial node or SA node, the pacemaker of the heart. Now, let's talk about the mechanism of conduction of cardiac impulse or electrical activities of heart by junctional tissues. So now let's start. Now look, what is junctional tissue of human heart? Cardiac muscle consists essentially of certain specialized structure which are responsible for initiation and transmission of cardiac impulse at a higher rate than the rest of the muscle. Those specialized cardiac tissues operate such mechanisms are collectively known as the junctional tissue of heart. The junctional tissue of heart made up of by the following structures. First, sinoatrial node. Second, atrioventricular node. Third, bundle of His. And fourth, Purkinje fibers. Now let's talk about the sinoatrial node. It is situated in the right atrium at the junction of superior vena cava and right auricular appendage. It extends downward along the sulcus terminalis for about 2 cm. It is broader at the top and trapping below and measures about 5 into 20 mm. Histology. It is made up of modified cardiac muscle fibers. The fibers are thin, elongated, trapping at the ends longitudinally striated and intercrossing with one another in a plexiform manner. The functions of SA node. It is generates the normal cardiac impulse at the rate of 70 to 80 per minute in the adult and acts as the pacemaker of heart, and the rhythm originated from this region is generally designated as sinus rhythm. Next, the atrioventricular node or AV node. It is situated in the right atrium at the posterior part of the interatrial septum close to the opening of the coronary sinus. It measures about 2 into 5 millimeter. The cells of the AV node are cardiac muscle fiber but have a few myofibrils. It also consists of Purkinje fiber which form a dense network. The node is extended into the atrioventricular bundle. The functions of AV node. First, it receives the impulse originated from the SA node and transmit it to the ventricles through the bundle of His. Second, it acts as a reserve pacemaker. The rhythms that is originated in the AV node is known as nodal rhythm. Third, it also initiates the cardiac impulse but at a lower rate, about 40 to 60 per minute. In abnormal conditions, when the SA node fails, the AV node generates the impulse or the nodal rhythm. Now focus on bundle of His. The main trunk of this bundle is continuous with the AV node and passes upward until it reaches the posterior margin of the membranous part of the interventricular septum and then forwards below it. It measures about 20 millimeter long. The functions of bundle of His. First functions is conduction. Its normal function is to conduct the atrial impulse to the ventricles, and the second role is rhythmicity. When the SA node and AV node fail, the bundle can originate cardiac impulse, but the rate is very slow, about 36 per minute. Next, the Purkinje fiber. The Purkinje fibers which arises from the branches of the bundle of His spread from the interventricular septum directly to the papillary muscle and then to the lateral wall of the ventricles, ending ultimately within the subendocardial network. Purkinje fibers have got a larger diameter than the ordinary cardiac muscle fiber. It also contains relatively more sarcoplasm with large amount of glycogen. Myofibrils in the fiber are present mostly in the periphery of the cell, and the central space is occupied by the glycogen. Now look at the function of Purkinje fiber. Main functions of those fiber is to conduction of impulse quickly to every part of the ventricle's muscle fiber. Those fiber also can initiate impulse 30 to 35 per minute in case of atrioventricular dissociation. This specialized conducting tissue, the Purkinje fiber, has been demonstrated only in the ventricles of the birds and mammals. In fish, amphibia and reptiles, those are not usually present, and conduction is made through the ordinary cardiac muscle fibers. Thanks for watching my video. Please like, share and subscribe the channel. Thank you friends.